Ever wonder how antibiotics actually work? It can seem like a bit of a mystery, right?
It really can.
Well, today we're going to be delving into a really important group, macrolides.
Yeah, a crucial class of antibiotics.
So we'll be going through a deep dive today and trying to really give you a good understanding of what these are, how they work, and why they matter.
Right.
And we've got a ton of great material about their structure, some well-known examples like erythromycin and azithromycin,
Yeah.
the tactics they use against bacteria, what they treat, the possible downsides to using them, and even how bacteria can become resistant to them.
Absolutely, a broad overview. Aiming for clarity and insight, giving everyone the essentials on macrolides without getting lost in the nitty-gritty.
So let's jump right in. What actually defines a macrolide antibiotic?
Okay. Well, macrolides are mainly bacteriostatic
Which
antibiotics.
means,
They mostly work by stopping bacteria from growing, and their defining characteristic is this large macrocyclic lactone ring structure they possess. They are vital alternatives for those who have penicillin allergies, and they're actually very effective against a lot of gram-positive bacteria and what we call atypical pathogens.
atypical pathogen. So what does that mean exactly? And what about this ring?
Ah, right. Well, for atypical pathogens, think about bacteria like Mycoplasma and Chlamydia.
Okay,
These differ in their structure or growth compared to the more common types of bacteria.
I see.
So they need macrolides for effective treatment. As for the lactone ring, it's usually 14, 15, or 16 atoms in size with deoxysugars attached.
Okay.
Imagine erythromycin. Our source material describes this ring as red with the sugars in blue. A nice visual.
Right, so I can kind of picture that now. Erythromycin is one example, but are there any other macrolides we should be familiar with?
Erythromycin is kind of the original. It works against things like streptococcus and Legionella,
Oh, okay,
used for a lot of respiratory issues and skin infections.
I see.
But one of the downsides is it can lead to stomach problems,
Right.
and it can interact with a lot of different medications
Why is that?
because it inhibits a key liver enzyme called CYP3A4, which is responsible for breaking down medications.
Ah, I
So
see.
that's erythromycin. Then you've got clarithromycin,
Okay.
a semi-synthetic version of erythromycin. It's much more stable, better absorbed. It can cover more bacteria,
Such as?
like H. influenza and H. pylori,
Wow.
and it lasts longer in the body, too.
That's pretty remarkable.
Fewer GI issues, but it can still affect CYP3A4.
Right, right. What about azithromycin? Everyone's heard of that one, right?
Yes, azithromycin is really widely used, often just a once daily dose. It's another semi-synthetic, but slightly different.
How so?
It has that 15-membered ring we were talking about.
Okay.
And it's excellent at getting into tissues.
Oh.
And it has a very long half-life. Azithromycin's good against a broader range of gram-negative bacteria as well.
That's interesting.
And less stomach upset and weaker CYP3A4 inhibition, so that means fewer drug interactions overall.
So fewer side effects, basically.
Exactly. There are others like roxithromycin, for example. And then there's telithromycin, which is part of a newer subclass called ketolides. It's got a really broad range of activity, but some potential liver toxicity concerns.
Right, right. That's really interesting. So you've gone through all these different examples, but how do these drugs actually go about stopping bacteria? Like, what's their mechanism of action?
Well, essentially, they interfere with the bacteria's protein production, which, as you can imagine, is absolutely crucial for them to thrive.
Right.
So these macrolides, they bind to what's called the 50S subunit of the bacterial ribosome,
Okay.
specifically to the 23S rRNA, which is near the exit site for new proteins.
So they're blocking that exit site.
Exactly. They block the protein chain from getting out. And, this disrupts that whole protein-making process.
So kind of jamming up the machinery.
That's a great way to put it.
That makes a lot of sense.
Yeah.
And this mainly leads to them, well, halting their growth.
Yeah, mainly bacteriostatic, stopping growth. But in high doses, they can actually kill some bacteria as well.
Interesting. So we've talked about how they work, but what kind of infections are macrolides typically used to treat?
Well, as I mentioned, they're great against a lot of those gram-positive bacteria, but unfortunately resistance is increasing.
Right.
It's still effective against those atypical pathogens we talked about as well.
Okay.
They're used for certain gram-negative infections too, things like whooping cough and some types of pneumonia.
I see.
And clarithromycin, that's the one that plays a key role in treating H. pylori, which is what causes stomach ulcers.
Oh, that's quite a range of uses.
Yeah.
So shifting gears a little bit, what are some important things to be aware of when macrolides are being prescribed,
Well, the common side effects are usually stomach issues, particularly with erythromycin, as I mentioned before.
right?
There's also a risk of what we call QT prolongation.
Which is,
It affects the heart's electrical rhythm.
okay.
And again, that risk is more pronounced with erythromycin. So, doctors have to be cautious when prescribing it alongside other drugs that have a similar effect.
I see. So they could compound each other.
Exactly. And as we already touched on, both erythromycin and clarithromycin can interact with a lot of common medications because of their effect on that CYP3A4 enzyme.
Right, right.
But azithromycin, luckily, has a much lower risk of those sorts of issues.
That's good to know. We've talked a lot about the positives, but what about this growing concern of antibiotic resistance?
Yeah, resistance to macrolides is a big challenge these days. Bacteria are crafty. They can actually develop resistance by modifying the part of their ribosomes that the macrolide would bind to.
So they can just change their structure.
In a way, often through genes, which can even give them resistance to some other antibiotics.
Wow.
Another way they do it is through what are called efflux pumps.
What do those do?
They actively expel the drug from the bacterial cell. This is more common with those 14 and 15-membered macrolides.
Okay.
And less frequently, bacteria can produce enzymes that break down the macrolide entirely.
So like actually destroy it.
Precisely.
That's pretty incredible, this constant battle that's going on. I find it fascinating. So we've talked about how they work, the problems that can arise. But how are these medications actually made in the first place?
Well, erythromycin, the first macrolide, is produced by fermentation using the bacterium Saccharopolyspora erythrea,
Okay.
but clarithromycin and azithromycin are what we call semi-synthetic. They're made by modifying that original erythromycin molecule.
Right, right. So to summarize, I think we've learned that macrolides are a really diverse and important class of antibiotics, vital for those with penicillin allergies and effective against specific types of infections.
Definitely.
And they work by inhibiting bacterial protein synthesis.
Right.
But it's important to remember that each macrolide has its own unique profile in terms of what it can treat, potential side effects, and the possibility of interactions with other medications. And this whole issue of antibiotic resistance is a major concern.
I couldn't agree more. Understanding these key differences and the mechanisms of resistance is so important for ensuring these drugs remain effective.
This has been incredibly informative. I've learned a lot.
Glad to hear it.
But this whole conversation has really brought up something kind of crucial.
What's that?
Considering how this antibiotic resistance is on the rise, what role do we each play in using antibiotics responsibly? How do we make sure that macrolides and other antibiotics remain effective for future generations? Something to think about.
Definitely something to ponder.
Well, on that note, thank you so much for joining me on this deep dive.
My pleasure.
That was fascinating.
It was.
And thanks to all of you for listening. Until next time.
See ya.